Hello, it's 4 o'clock and welcome to Biology 107. So welcome to class. Today we're going to cover the course outline, talk about a few other things. We're going to get into some of the lecture materials and tell you all about this class. So let's get into things and I'll tell you all about it. So first, this is me. My name is Blaine McGarry. I have a PhD from the University of Guelph, and my background is microbiology and biochemistry, and maybe I'll say a little bit more about that in a minute. Currently, the college has switched to online learning for lectures, and the labs will be in person. So I'll talk a little bit more about the labs in a few minutes. So for the time being, you're going to be attending lectures on Zoom. Obviously, you figured that out since you're here, and hopefully in a few weeks we'll switch back to in-person. In-person is just so much better. I do have office hours, but currently I'm kind of working at home some days and working at the office some days, so you can try to drop in. If you need to see me, I may or may not be there. At the moment, the best way to get a hold of me is by email or book an appointment. And I can set up something for in person, or we can set up something for in Zoom. You can try phoning, but since I'm working at home, I'm probably not going to be in my office a lot of days. So you can worry about that by emailing me. Before we get into things, I just want to talk a little bit about Zoom etiquette. Kind of just general classroom etiquette is being on time and being respectful. Don't need to say much more about that. One thing that's important to tell people is please dress appropriately and be aware of your surroundings. I do have Zoom set up so that your camera and your microphone are turned off automatically, but I don't want to have there be any accidents where you accidentally turn on the camera and you're in your underwear, or who knows what, maybe someone in your background is in their underwear, or, I mean, I don't need to go there, and I don't need you to be going there either. So please dress appropriately and come prepared for class. Your microphone will be muted. Just keep it muted unless you're asking a question. If you need to ask a question, feel free to unmute it. Just, sometimes people, dogs barking in the background and other people in your house clattering around. We want to kind of minimize that kind of thing. You can ask questions using the chat function on Zoom. In the Zoom bar, there is a chat function. You can just type in questions or whatever there and interact with the class that way as well. And of course, come prepared to learn, because that's the way you're going to get the most out of this class. As mentioned, we are going to be doing labs in person, which is great. And we will be following all the Keanu COVID protocols. So I think everyone's probably familiar with these if you've been on campus all semester in the fall. It's obviously a changing situation. If there's anything new, I will inform you, and you'll probably hear it from the college as well. So just keep in mind, we will be following all the COVID protocols. So a little bit more about me. As I mentioned, I have a PhD from the University of Guelph, and my background is bacteria and cell biology. And so this is the right place for me to be in terms of teaching this course. There's some pictures of, these are my mutant babies, I call them. It's working on cell wall synthesis in bacteria. And some of the mutants that I had made actually changed cell shape, which is kind of interesting. Maybe I'll say more about these another day, maybe not, I don't know. But if you're interested in these things, you've come to the right place. Okay, so let's talk about this course. We're going to talk a lot about cells, obviously. It's a cell biology course. We're going to talk about structures of cells, organelles. We're going to talk about energy and matter. We're going to talk about viruses. We're going to talk about, we have a lab section, so we're going to learn a lot of lab techniques, particularly microscopy. And along with those labs, you're going to be doing a lot of technical and scientific writing. And that's going to be a big part of the course. And I'll talk about that in a few minutes here. Some things you're going to need for the course. We do follow this textbook here. This is Campbell's Biology 3rd edition. There's other editions out there that are perfectly acceptable. If you're not sure, just let me know. This is pretty big, heavy textbook, but if you're doing any other biology courses, Biology 108, you're going to need it for that. This is, in my mind, one of the better biology textbooks out there, but there are many out there. If you have something else that works for you, that's fine. You're going to be tested off my notes, but like I said, this is a very good textbook, and I do recommend getting it and reading it, but not 100% essential. You can always, as well, get a digital version of it as well. And like I said, there's many biology textbooks out there. The introductory ones are all very similar to one another in terms of the content and whatnot. If you're getting anything that's a little old, let me know, and I can see if it's how relevant it is or not. But I do accept the last couple of editions as well. I know they like to change them up quite frequently. So other things you're going to need a lab manual, and you can get that at the bookstore. I think, I'm just trying to remember, I went to the bookstore the other day and I think they had them in stock. You're gonna need a lab coat. Your lab coat is gonna be stored in a Ziploc bag for transporting it between the lab and other places. You're going to need a permanent marker, and don't be that person that's always borrowing one. Go buy one. You can get them at the bookstore or just go to any other place. You're gonna need them for labeling test tubes and whatnot. Please make sure you have one. And lab goggles, we really only need them for one exercise in lab 3. So if you don't have lab goggles, that's fine. We probably, between everyone and their partner, there will be enough pairs. If you're concerned about sharing infection and whatnot, then we can certainly sanitize them after and or before use. But if you have them for chemistry or something like that, then you're probably set, but really only need them for lab 3. If you do feel you need them more, sometimes there are other exercises, but in a lot of cases we're working with plants and water and harmless things that aren't going to give you any eye damage if they're put in the eye. It's lab 3 where we have some solvents we're actually dealing with. So let's talk about the evaluation of the course. There are 2 midterms and a final exam. So you can see that's 15, 15, and 30. So that's 60% of the course. So you are going to be able to, you are going to need to be able to write tests in this course. And we're going to talk about tests, and I'm going to try to give you some tips on writing tests as we go along and go through the course material. We'll get there. Some of the tests are going to be a combination of multiple choice and written, and sometimes problems. And like I said, it's important to pay attention in class when I talk about those things and give you ideas on how to approach the test questions. And we'll try to spend a bit of time on that this semester to coach you through these things. The lab is a big chunk of the course. That's 40%. I'm going to talk about the lab here in a moment. I have the lab schedule and I sent it to you the other day. So I'll come back to the lab in a second, but that is a big chunk of the course. This is the evaluation scheme for the course. If you've been around the college before, then you probably know we use a letter grade system, and there's the conversions. Most places for transfer are looking for a C- minimum. U of A is anyway. If you go to other institutions, they may have different criteria. So we take the U of A standard in terms of what we say is the minimal, I guess you'd say passing grade, 60. But I'm hoping that people are hoping to achieve better than 60. So in terms of the course content, this is kind of the format. We have the first chunk of the course, we talk about different cell structures, so organelles, the 2 types of cells, eukaryotes and prokaryotes. And I have a unit on viruses and COVID-19 that technically the virus unit is topic 20, but this last year I've decided to put it kind of up at the front at the beginning because it's been extremely relevant to what's going on in the world today. So I'm going to put that in, maybe I'll put it around topic 3 or topic 4. I haven't quite decided where it's going to be. We're going to cover that material, and then we're going to have our first midterm on February 11th, and that is before the reading break. The second part of the course, we're going to talk about energy and processes such as respiration and cell division and things like that. And we'll have another midterm on March 18th. And then the last part of the course, we're going to talk about DNA and information and all of these processes, replication, transcription, and translation, and a little bit about DNA technology and forensics and things like that. And then we'll have our final exam. The date is not set yet, but that will be sometime in mid to late April, whenever the exam period is. So one thing you're going to need for the course is Moodle. I'm going to actually go to the course Moodle page here right now. And let me just change my screens. There we go, and just show you a few things. So here's the course Moodle page. I have a ton of stuff here. So I'm just going to scroll down and point out a few things. First, if you don't need the email to get to the Zoom link, I put the Zoom link here for lectures. And all our Zoom lectures will be recorded. So I will post a playlist here of our recorded lectures and any other materials that are kind of important for the course. We have a WMIS course that you need to take. If you've done it already, you can upload your certificate here. We also have a plagiarism recognition tutorial course, and you can upload your certificate here. So those are two courses that you can do this week. We have no lab tomorrow. So tomorrow afternoon is a good time to do these courses if you have not done them already. If you already have the certificates, upload them, and that's all good. And I need this safety course done before you go to the first lab, and I need the plagiarism tutorial certificate done before you hand in any assignments. So the sooner you do that, the better, and you might as well get it over with. You can see I have lecture notes down here. I have two versions of my lecture notes. I have these PDFs. I'll click on one and show you what they look like. And where did it go? I guess it downloaded the file. So let me just pop that up here. So here's what my PDFs look like. There are a lot of words, a lot of written information. You can see there's a little, some few pictures here and there. And these are all available before the lectures happen. And the PowerPoints are mostly pictures. And the PowerPoints sometimes they're available beforehand. I try to make them available beforehand if I can. And probably I'll be able to do that. But if not, they will be available after the actual lecture. But they follow each other basically beat for beat, just that the notes have, like I said, are more words and written materials and information and questions and definitions and those kind of things, whereas the PowerPoints are more pictures. The laboratory schedule down here, and you can see right here, hold on a second, I guess I'm not back to here we go, sorry. There's the laboratory schedule. And you're going to be handing in your lab reports on Moodle. So you can see here's the link for the first one. That's due January 23rd. Hopefully I have that date correct. I'll double check that before it happens. And eventually I'll have some more lecture notes and other things there, review materials and whatnot. So you'll be probably using Moodle a lot for this course. So hopefully everyone can find their way there and get all the notes and everything else. And if you have any questions, please let me know. So a bit more about the labs. Here's the lab schedule. So labs start next week. So that is Thursday, January 13th. And so we'll start in lab 1. So make sure you have your safety course done before then, or you won't be allowed in the lab. You'll make sure you have your lab coat and those kind of things. And so you can see kind of the format of how the labs work, is we have labs 1 and 2, and then you're going to complete those 2 labs, and then you'll have a lab report that will be due the following week. So I think actually I have the dates wrong here. This should say January 27th. So I'll have to update that and fix it for the, yeah, it should say the 27th. I probably copied and pasted that from something and didn't change the due dates because it'll be due 1 week after lab 2. Lab 3, if you take a look, it shows the percentage of what they're worth. So the first one is what I call a short report. And then the second one here for lab 3 is a formal report, and it's worth quite a bit more. And you're going to see it's quite a bit more work. I'm going to give you 2 weeks to do that one. Other short reports are worth 3 or 4%, and we'll talk about report writing as we go along over the semester. At the end of the semester, there's a microscope test. So that will be March 24th, and there's a lab exam as well. And we'll talk about those things as we get closer to the actual dates. So this is just a reminder to do the safety course. The safety course includes WMIS, biological safety, and a few other things. And it's required for taking any labs in this department. So make sure you do that right away and get your certificate uploaded to Moodle. Now, if you're taking any other lab courses, your instructors will be asking for it as well. So one thing just to note is Our lab is a biosafety lab, meaning that we do work with microorganisms, bacteria that are potentially infectious. The risk is extremely low, but there is a small amount of risk. So we do have to follow certain rules that are laid out by the Public Health Agency of Canada. And most of these are pretty straightforward, kind of common laboratory rules, like no food or drink, make sure you wear your lab coat, washing your hands, and I'll be reminding you of these rules as we go along. If you have any bags or jackets, they're not allowed in the lab. We don't have any storage area for them in the lab. And so you'll have to leave them in the hallway or your locker or car or wherever, whatever works for you. What to bring in the lab? Your lab coat in a big bag. If you don't have a big Ziploc bag, you can buy them at the college bookstore. Your lab manual, pen, I guess I should add a marker on that list. Things to leave behind, your jacket, your bags, those kind of things, food. Cell phones are allowed, of course. I'm not going to ask you to leave them out in the hallway unguarded, but anything like that, if you're going to take it out, you just got to make sure that we're cleaning it off if it gets contaminated. So it's just kind of a reminder there to, make sure we keep our things clean. Yeah, just a reminder, leave your extra stuff behind that you don't need. We also need proper footwear. This is usually not a concern in the wintertime. People are covering their feet, but in the fall time, people are trying to wear flip-flops and things like that. And so we want to make sure we are having footwear that covers the foot. So those ones there you can see do cover the foot. I probably don't recommend this boot kind of thing. You want something comfortable. You're going to be on your feet for 2 or 3 hours. So you do want to have something relatively comfortable. Another thing is we do have to cover our ankles. A lot of people like to wear the short socks. So make sure you wear, pants that go all the way down or full socks or something like that. And we'll be in good shape. Okay, so just some tips on success. And I want to talk a little bit about studying and things like that. And we'll see if we get into that first lecture or not. But first, come to class. I could probably make a graph like this for a lot of semesters in terms of attendance and marks. And there is a bit of a correlation between those who come to class and engage with material and those who don't. Come prepared to learn. Hopefully that goes without saying. Ask questions, engage, do the assignments. Every semester I get some people who don't do the assignments and they are worth marks. And I had one semester somebody who didn't do any of the lab stuff. That's 40% of the grade. So I don't know how she expected to pass the course when she's ignoring 40% of the course. That was really unfortunate. Take good notes and review them. And I'll talk about note taking here in a moment. I've got a little video to share with you. And learn effective study methods. Some people come to college and they know how to study and some don't. And there's lots of resources out there on the college and other places that can help you with these things. And that's what I actually have a video to play for you here that talks a little bit about studying biology. So this is a pretty short video. I can't remember, it's 2 to 3 minutes. And it talks about a good way to study biology. So I encourage you to take some of this heart because I think it's got some really good advice here. So I'll play that for you right now. Okay, so that is the end of the video, and you can see they're just talking about taking notes and why take notes. You're trying to remember stuff, and if you're just passively listening or just passively watching videos, how much do you think you actually remember? Even just taking notes and not reviewing them again, you actually double the amount that you learn. So if you're only learning, remembering 5 to 10%, you can already double that to 10 to 20%. Taking notes allows you to structure things, organize them, and put them in your brain and help to form those long-term memories. Even better, teach something to your friend. That forces you to organize it. It forces you to understand it before you can get it in your brain. If you have any questions about note-taking and those kind of things, another suggestion I always say is talk to your friends and find out what study methods they are doing, what works for them and what doesn't work. And this is how you're going to learn how to study. We also have lots of resources at the Academic Success Center, and I encourage you to look into those things as well. So this is just my big plug for why we should take notes. Like I said, helps with your memory, helps with studying, and if I'm giving you tips or hints about the exams, then it's always good to have those things for review. Another thing just to consider is, I went to this workshop. They were talking about this research done on students and technology, and these are good questions. How much time are you spending on these things? Probably not 8 hours a day, but they're probably a distraction. And so I encourage you, when you're in class, put these things aside, deal with them later, focus, come to class, come to learn, those kind of things. So one other thing we're going to do this semester is we're going to do some Kahoots. So we're not doing one today, but we'll do one next day. A Kahoot is it's an app that you can download. So I encourage you to download it for Friday. And what is it? It's kind of like a little quiz show, just to break up the lectures and maybe add a little fun and test yourself. And so download the app, and we'll see what they're all about on Friday. If you don't want to download the app, that's fine. You can always go and use a web browser as well for Kahoot. Okay, so a couple other things just to finish up, and then I want to, like I said, get right into topic one. This one here is just for fun. I wish, but I guess that's not a real thing. That's just kind of a little joke. Another thing is, don't forget to take a look at the entire course outline. It's always worth to flip through it, see if there's anything important that I've missed. And it is a document that is, we're supposed to follow as part of the college and make sure that we are covering all that material. So take a look at the course outline. There may be things in there for you. Let's get into topic one. Today, I just want to do a quick introduction to topic one. And topic one is a little bit of this, a little bit of that, all about life and the cell and the scientific method, and biology and things like that. So I just want to kind of get into this and get going with the lecture material and start thinking about biology right off at the beginning. So here is planet Earth, and we are somewhere around there, somewhere in North America. And so what is biology, right? Biology is the study of life. And one way to think about biology is a hierarchy of biology. You could talk about the whole thing, which is the biosphere, the planet, and all the living organisms on it, and all the living organic matter. Sometimes we talk about smaller things, ecosystems or communities, populations, organisms. And actually, this slide here is a little bit more about what we would be talking about in Biology 108. So Biology 108 is, I think it's called Introduction to Biodiversity. So if you take Biology 108, you're going to talk about a lot of this stuff, different types of organisms, plants and fungi and animals and all sorts of things like that. Biology 107, we're going to zoom in, and we're going to talk more about cells, and we're going to talk about organelles and molecules and all of those things and how they work and how they put together the whole thing. So this is where we're going to focus in on cells. So biology is the study of life. What is life? It's a great cereal. I used to eat that a lot when I was a student. I discovered it when I was in university. I didn't know about it before. But I want to talk a little bit about what is life, because it's kind of worth it to think about what is living in general. One way we can talk about life is the characteristics of life. So what do living things have? They have order and organization. So you can see that's very obvious in some organisms, such as the sunflower. There are non-living things that have order. You think about like a salt crystal or something like that. So life isn't just something that's organized. It actually has a number of different characteristics. So reproduction, another characteristic of life. You can see there's a great Canadian animal there. Growth and development. So growth and development, hopefully that one's pretty obvious what that is. Energy processing. This is something that distinguishes us from other organized things, such as salt crystals that can grow, but they're not processing energy. They're not changing molecules from one form to another. So we're going to talk a lot about energy processing this semester. Response to the environment, homeostasis and regulation. So these are all things that are part of life. Evolutionary adaptation, meaning that organisms' offspring, some of them survive and some of them don't, and the ones that survive are more likely to propagate and have their own offspring. So what is a cell? This is the next question to consider, right? If a cell is alive. A cell is, of course, the basic building block of all living things. There's different ways we can kind of define cell, but this is kind of one of the better definitions, right? It's a basic unit of life. And it could be one organism, or it could be part of one organism. So there's a lot of organisms that are single cellular organisms, so such as bacteria. One organism is one cell. Other organisms, we could have many cells. So the human body is something like 30 to 50 trillion cells. And so cells are part of us, they're just building blocks in our case. Cells are the basic structures that provide a whole bunch of different functions, such as structure, energy metabolism. They carry our genetic material and many, many other things. So we're going to touch on a lot of these things over the semester. There are, of course, many different types of cells. This is not really a physiology or anatomy class. So we're not going to get into all these different types, but we're going to focus on some of the basic types. We're going to talk about bacterial cells. We're going to talk about animal cells and plant cells. Those are kind of the three biggest ones we're going to talk about a lot. Some of the differences in terms of structures and function. We'll talk a little bit about protists and a little bit about fungi and a few other things like that. We're going to over the semester. And like I said, one thing that's really important about cells is they carry our hereditary material, which of course is DNA. And we're going to talk a lot about DNA in the last third of the course after midterm number 2. So are cells alive? Yes. Remember the criteria or characteristics of life. Cells have order, they reproduce, they grow, they utilize energy, they adapt, they respond to the environment, all those kind of things. And so, like I said, we're going to talk about the animal cells and bacterial cells are going to be one of the two of the main types we're going to talk about during the semester. An animal cell is also known as a eukaryote. So I will write that out for you. Eukaryote. And a bacterial cell is a prokaryote. And we're going to get very familiar with those words over the semester. We're going to talk lots and lots about the differences between these things and what they can do and why they're so different. So something else we're going to talk about are viruses, and little bits in here and there. We have one topic on viruses, and then they sort of pop up here and there over the semester. Of course, this is maybe the most famous virus of the year, and it's hard to believe that 2 years ago it hadn't even been discovered. It was discovered 2 years ago in January, around 15th or something. It was isolated, but so kind of hard to believe that at the moment. What is a virus? So more on these when we get to that topic, but basically a virus is a piece of nucleic acid so DNA or RNA, and it's surrounded by a protein coat. Sometimes it has other features on it, like it might have a membrane or something else, but that's basically what it is. So it's instructions that are packaged up in a protein. This is what the SARS coronavirus 2 looks like, and in terms of its structure. And of course, everyone's talking about the spike protein because this is what the vaccines are based on, is the spike protein. So, more on viruses, but basically they're infectious particles, and they're made out of instructions surrounded by proteins. So, question is, are they alive? This is kind of one of these things that people kind of debate a little bit. Most people would say that they are not alive, although some people kind of debate that a little bit. If you take a look at the characteristics of life, you can sort of check off some boxes, right? Is there organization there? Well, yes, there's definitely organization. You've got these instructions. They're surrounded in a protein coat, so they're organized. Do they reproduce? Do they grow? Sort of. They need a cell to do this, right? So we could put that they need a cell host in order to do this, which is kind of different than doing it yourself, right? Do they utilize energy? No, they do not. Do they respond to the environment? Not really homeostasis, but they do adapt. They certainly do. In case you haven't been paying attention to the news, where you get these new variants that pop up, and they adapt, and the best, most efficient virus wins, in a sense, the one that survives the most. So are they alive? Usually we don't say they're alive. A lot of people would say they're not, but some people would say that they're sort of alive or something like that. So, I'm sure we could have a debate over it if you wanted to. So cell biology is a study of all sorts of things, different types of cells, cells, animal cells, protists, bacteria, fungi. I think I mentioned all those before. We're going to zoom in. We're going to talk about cellular components, so organelles and cytoplasm and structures that help these things swim, called flagella. We're going to talk about molecules, proteins, nucleic acids, and so on. And we're going to talk about viruses. And there are lots of other things we could talk about. And cell biology is one of these branches of biology that it does span many sort of subdisciplines. You could consider genetics and biochemistry, and all sorts of other disciplines are sort of, in some big Venn diagram of studying biology. So we are going to talk about molecules. That's why you need Chemistry 30 as a prerequisite to this course. So a little bit of chemistry here and there we're going to sprinkle about, and these things and many, many more. So this will be my last slide. I'm just going to show you a little tool on the internet, and then we'll wrap it up for the day. But just showing you that kind of cell biology, if you think about it, sort of zooms in on this area. And if you take a look at the bottom of this diagram, most of these things require a microscope in order to view them. And so we're talking about microscopic things, which I think are super, super fascinating. So I wanted to kind of finish off today and show you this tool that I found on the internet. It's called The Scale of the Universe. I think you can download an app as well. And it's kind of just something I like to show people because it's really fun. I'm just going to change my web browser because here it is on the internet. And I'm going to hit start, and I'll show you what this is. It's really, really interesting. Maybe I'll turn off the music. It's a little annoying. So if you take a look at this, it's something where you can kind of zoom in or zoom out, those kind of things. You can see there are all sorts of different objects there. There's a human. You can click on the human, and there's a little blurb. Sometimes the blurbs are informative, or they're cute or funny. And like I said, this is one of these things I get stuck in sometimes, just sort of playing around with because it's lots of fun. He says, I'm going to assume you are human too. Probably a good assumption. You can click on some of these other things, like a sunflower. Look at that. Sunflowers are taller than humans, or they can be anyway. That one says about 2.5 meters. And it says it's actually made of thousands of small flowers. So I didn't know that. You can zoom out. You can see we've got animals that are extinct and otherwise, some large ones. There's got to be a blue whale here somewhere. There it is. because everyone knows blue whale is the biggest animal that ever lived. Apparently 180 metric tons. That is very large. Lots of other things, human-made and otherwise. You can see there's some human-made structures, the Great Pyramids, some natural things, the Half Dome in Yosemite. I don't know if Niagara Falls is in here. It'd be nice to see some more Canadian content, of course. Halley's Comet, Mount Everest. You can zoom right out and you can keep on going. You can see that we've got different countries and moons. And like I said, this is kind of a lot of fun. There's Earth, and you can see how Earth is very similar size to Venus, and Mars is about half the size or something like that. And this massive continent here, Asia, is huge. I don't know what Sirius B is. It looks like it's a white dwarf. There's the Minecraft world. You can zoom way out if you want and check out these things, all sorts of nebula and stars. But what I want to do for this class is just zoom in a little bit to some teeny tiny things. So there's the human, and here we go. And these are the things that we care about in this course. So there's some organisms, matchstick, those kind of things. And here we go, and now we're getting to duckweed. So the duckweed is a very small plant. And if you take a look at this, we're looking at some very large cells here. We're actually going to be talking about paramecium and looking at paramecium in Lab 5 this semester. It's a relatively large protist, about 200 micrometers, and they like to swim around. You can see this little thing here is human hair. And a human hair is worth mentioning because that's about the smallest object we can see in terms of the width. So everything smaller than this, we need a microscope to see. So you can see we're talking about white blood cells, cell nucleus, chloroplast, red blood cells, bacteria. There's E. coli. Get to this range here, and suddenly we're looking at things that you can only see using an electron microscope. So we're looking at viruses and DNA and other molecules. So I encourage you to check it out. Like I said, it's a lot of fun and kind of just worth a click around because these things are pretty cool. But that is where I wanted to finish off today. So on Friday, we're going to pick up where we left off and get right back into topic 1. We're going to talk a little bit about a history of cell biology and get into things like the scientific method and whatnot. And so hopefully a lot of little kind of review, maybe a few new things in there for you and some interesting things. And then next week, we're going to get really thick into the course and into the content.